This lecture covers osmoregulation and excretion. The role of the excretory system, also called the urinary system, is to eliminate body wastes. The principal wastes produced by the body are called nitrogenous wastes, and these are the breakdown products of both proteins and nucleic acids. But the excretory system also has a crucial function in ensuring fluid balance. Water in the body resides in two different compartments. We have the intracellular compartment and the extracellular compartment, which includes the interstitial space and the intravascular compartment. The movement of water from the intracellular compartment to the extracellular compartment is dictated by the amount of solutes dissolved within the intracellular compartment versus the extracellular compartment. The excretory system therefore maintains fluid balance by secreting excess solutes in the urine. In other words, the excretory system and osmoregulation are inextricably linked. Our first learning outcome is to therefore define and illustrate what is osmoregulation. Our second learning outcome is to describe the different forms of nitrogenous wastes and why the animal kingdom needs different forms of nitrogenous wastes. That is our second learning outcome. Our third learning outcome is to identify and compare the diversity of excretory systems within the animal kingdom, followed by an in-depth analysis of the mammalian excretory system. Osmoregulation regulates osmosis, as its name implies, osmoregulation. So osmoregulation is the regulation of osmosis, and osmosis is the movement of water across a semi-permeable membrane. And so regulation of osmosis is achieved by maintaining electrolyte homeostasis. The electrolytes are minerals that we obtain from the foods that we ingest. Some of these electrolytes end up in the extracellular compartment, while others reside in the intracellular compartment. The extracellular compartment can be further subdivided into the intravascular compartment. This is inside blood vessels, the intravascular, and the interstitial fluid compartment. Because they reside outside of body cells, we also group them as extracellular fluid compartment, the extracellular fluid compartment. And this, of course, is our intracellular fluid compartment. Sodium is our most important extracellular electrolyte that governs osmosis. So we find sodium in the extracellular fluid compartment. And it's largely responsible for the movement of water between the intracellular and the extracellular. Too much sodium leads to an increase in the volume of the extracellular fluid compartment. And so after a salty meal, you can feel your hands and your feet swelling, especially the morning after a very salty meal. Chloride and calcium ions are also important extracellular electrolytes. So we also find calcium in the extracellular fluid compartment and chloride ions. These play an important role in neuronal signaling and are not so important in affecting osmosis. Within cells, our principal intracellular ion is potassium. And so this is our principal intracellular ion that governs osmosis. Magnesium also plays a role, and so magnesium is also an intracellular electrolyte that is important for governing osmosis, although your table here does not indicate a role for magnesium as affecting fluid balance. And so just to recap, potassium we said was crucial in water balance, as was chloride. So both of these are crucial for regulating osmosis. In the excretory system, as we will see towards the end of this lecture, the nephron determines how much potassium is excreted in the urine, or how much is retained, how much is reabsorbed out of the filtrate, and also how much sodium is excreted in the urine. And so this interplay between sodium and potassium ensures that we maintain fluid balance, that we have the right volume of fluid within body cells and the right volume of fluid in the extracellular fluid compartment. Too much fluid in the extracellular fluid compartment would increase blood pressure, which means that the heart has to work even harder in order to maintain that pressure gradient in order to enable bulk flow. The interplay between the three main electrolytes that govern fluid balance, namely sodium, potassium, and magnesium, is captured in what we call the DASH diet. The DASH diet is an abbreviation for Dietary Approaches to Stop Hypertension. And the DASH diet recommends that we increase our potassium intake and magnesium intake. And increasing our intake of foods that have significant amounts of both magnesium and potassium can help lower blood pressure, even if we do nothing to reduce the amount of sodium. Ideally, we would also reduce the amount of sodium in the diet, and that way we can further decrease blood pressure. While we alter our electrolyte composition by eating, the excretory system then carries the burden of reestablishing electrolyte homeostasis. And at the end of this lecture, we will see how the kidneys secrete or recapture electrolytes from the filtrate that becomes urine in order to osmoregulate. Clearly, osmoregulation must come at a cost in the currency of ATP. Recall that our basal metabolic rate is the amount of energy the body consumes at rest. At rest, our kidneys continue to eliminate wastes and to osmoregulate. If we compare the kidneys to other major organs, the kidneys consume only 10% of our daily energy expenditures. And this might seem small compared to, say, the liver, brain, and the skeletal muscle tissue. But consider that a kidney is a mere 160 grams compared to the liver and the brain that are over 1 kilo. So the brain too is over 1 kilo. So for its size, the kidney is the most metabolically expensive organ to maintain. So what determines the cost of osmoregulation? There are three things that determine how much ATP is required for the osmoregulatory apparatuses of the body. The first is how different an animal's body, an animal's osmolarity is from its surrounding environment. For example, the osmolarity of the ocean is a staggering 1,000 milliosmoles per liter. And this unit tells us the amount of dissolved solutes within a liter of water. So there's a significant amount of solutes dissolved within a liter of seawater. Compared to the osmolarity of, say, marine fish, the osmolarity of marine fish ranges between 225 to roughly 400 milliosmoles per liter. And so the closer the osmolarity of an animal's body to its environment, the less energy the animal has to expend in order to maintain this gradient. The greater the difference, so an animal whose the osmolarity of its blood is 225 versus that of its surrounding environment being 1,000 milliosmoles, is going to have to spend a lot more ATP in order to maintain this gradient. Second factor that determines the amount of energy consumed in order to osmoregulate is how easily water or solutes move across the animal's body surface. In other words, how permeable the skin is to both solutes and water. And the more permeable, the more work is involved in osmoregulating. The third factor is the amount of ATP required to operate the active transport pumps embedded in the plasma membrane of cells in order to move solutes against their concentration gradients. So now that we have some appreciation of the cost of osmoregulation, one wonders, where do we find osmoconformers in the animal kingdom? So animals that don't osmoregulate, because this would imply that they require far fewer calories in their diet if they don't need to continuously osmoregulate. The simple rule for osmoconforming is largely set by the environment. All freshwater animals are osmoregulators. They have to osmoregulate because the solute concentration of fresh water is simply too low to allow the neurons to fire action potentials and for muscles to contract. And therefore, all freshwater animals are osmoregulators. All terrestrial animals are also all osmoregulators. Terrestrial animals consume food and drink fluids and therefore can easily disrupt the balance of internal electrolytes. And therefore, all freshwater animals and all terrestrial animals are all osmoregulators. We find osmoconformers exclusively in seawater, in marine environments, because the osmolarity of seawater is nearly always the same. It is constant. And therefore, this enables marine invertebrates and several marine vertebrates the option to osmoconform. So most marine invertebrates are osmoconformers because they lack fancy osmoregulatory apparatuses to osmoregulate, and because their surrounding environment doesn't fluctuate much, they are able to osmoconform. We also have some vertebrates marine vertebrates that are also osmoconformers. Again, because the environment, the salinity of the environment doesn't change much from one moment to the next. But there's an important caveat here. And the important caveat is that while these marine vertebrates have the same osmolarity as the surrounding seawater, which is roughly 1,000 milliosmoles, the composition of the electrolytes is different. And so they have a different constellation of electrolytes than what you would find in the surrounding ocean water. Let's look at one example of osmoregulation in marine birds. These marine birds are terrestrial animals that obtain their food from the sea, and this is why they are so interesting. These very large birds with impressive beaks gulp up a significant amount of seawater as they nosedive into the water and fish out their prey. Before the discovery of these salt glands that sit on top of the birds' heads, biologists assumed that these marine birds simply would spit out most of the seawater that they ingested. What they didn't know was that these peculiar structures that sit on top of their heads were assisting greatly in osmoregulation. So in addition to having a kidney system, the excretory system, they had an additional apparatus on the top of their head that worked hard, diligently in osmoregulating, in removing all of these excess solutes that are consumed every time they would swoosh into the water. So an artery within these salt glands as we see here, conveys blood down this network of tubules. The artery branches into a set of arterioles and eventually into a set of blood capillaries that are in intimate association with these excretory tubules. Solutes contained within the blood, so if we were to draw this out here, solutes contained within the blood diffuse out into the interstitial space and into these specialized cells called transport epithelial cells with a brush border, and this brush border increases the surface area through which all of these salts, these electrolytes, can diffuse quickly into these epithelial cells with a brush border and into the lumen of this tubule. And so then the flow of this fluid is eventually out from the nasal cavity at the top of the beaks here. And so we have this constant diffusion of blood with high osmolarity, diffusing down its concentration gradient into the interstitial fluid, and from the interstitial fluid diffusing into these transport epithelial cells. As the fluid works its way down, the osmolarity of this fluid continues to rise, and it continues to rise because we have a constant diffusion of solutes into the lumen of these tubules. But as the fluid works its way down, it comes into contact with blood with an ever increasing solute concentration. So we're always moving from a high concentration gradient here to a lower concentration gradient here. So net diffusion is always into the fluid contained within these tubules and eventually out through these nasal passages. And so you might have recognized this as another example of a counter-current exchange system. Fish also operate transport epithelial cells within the lamella tissue of their gills. In preparing for this lecture, I found this really nice quote from the intro of a paper that looked at these exchange surfaces. The fish gill is arguably one of the most complex transport epithelia in the biological world, where a single epithelium, so a single epithelial cell, as like the one that we just saw, is designed to simultaneously transport respiratory gases, to maintain electrolyte homeostasis, and in removing nitrogenous wastes. So to achieve this diversity in function, the epithelium covers hundreds of filaments that comprise roughly 50% of the total surface area of the animal. So contrast this role of these epithelial cells of transporting respiratory gases, maintaining electrolyte homeostasis, and removing nitrogenous waste, and contrast this for a moment with the epithelial tissue lining the inside of your lungs. Now all of a sudden your lungs seem quite lazy, performing only a singular task, which is to allow oxygen to diffuse in and carbon dioxide to diffuse out. These challenges are met, the challenges of these epithelial cells are met differently for freshwater fish versus marine fish. For marine fish, the main challenge is losing water by osmosis through the gills and other parts of the surface of the body. So as blood courses through these blood capillaries, we have a loss of water into the surrounding environment, because remember, the osmolarity of the surrounding seawater is 1,000 milliosmoles versus, say, roughly 300 milliosmoles inside the fluids of this fish. So we have a net loss of water to the environment. So as the blood gets oxygenated and carbon dioxide is eliminated, we also have the problem of losing water, significant amounts of water through these gills. The solution to this osmotic water loss to the environment is to produce scant amounts of urine. So very little urine is produced by marine fish. Also, to compensate, marine fish have to drink a significant amount of water to compensate for all of the water being lost through osmosis. The downside of drinking all of this water is the extra burden now placed on the transport epithelial cells studding the lamella here to excrete the salts out of the gills and into the surrounding environment against its concentration gradient. Recall that the concentration gradients of all of these solutes is very high on the outside, so this takes a significant amount of ATP to operate the transport pumps on the transport epithelial cells in order to move all of these solutes into the surrounding seawater. Freshwater fish have the opposite problem. They have a net uptake of water through osmosis from the surrounding water into their gills. If this went unattended, freshwater fish would eventually swell and burst. So to osmoregulate, the excess water is eliminated through copious amounts of water in the urine. So the significant amounts of urine are produced, which is a major difference from the marine fish. Freshwater fish also don't drink water, as do the marine fish, so there's no intake of water through drinking. And the expensive active transport pumps that stud the transport epithelial cells of the gill filaments work continuously to uptake salts from the surrounding water and move them against its concentration gradient into the through the gill capillaries and into the animal's body. This brings us to our second learning outcome, the elimination of nitrogenous wastes. Why are nitrogenous wastes so problematic? Recall from earlier lectures that energy-yielding macronutrients include carbohydrates, lipids, and proteins. Excess carbohydrates are stored as glycogen within our muscle tissue and fat cells. And so we can use this as a source of energy at a later time. But because we have a limited capacity to store glycogen, because glycogen is a very bulky molecule, the bulk of carbohydrates consumed are converted to fat and stored in our adipose tissue in the form of triglycerides. So they're stored as fat in our adipose tissue. Excess fat is also stored in adipocytes, our fatty tissue. So it's also stored as fat in our for later use. In the end, there are no waste products that are generated from carbohydrate and lipid metabolism other than the carbon dioxide once they are catabolized in order to generate ATP. So our only waste product is carbon dioxide. But the same no waste principle does not apply to proteins. Excess proteins must first be deaminated, meaning the amino group here must be eliminated so that the carbon skeleton can be converted to fat and metabolized for energy. So proteins too can be stored as fat and metabolized for energy. So the waste product here would also be carbon dioxide. But before being converted to fat, the amino group has to be eliminated. So we have a waste product, and the waste product is ammonia. This clipped off group then, if not eliminated, can alter the pH of our fluids and disrupt the three-dimensional shape of all body protein. And so ammonia is highly toxic. The amino groups from amino acids and also from the nitrogenous bases shown here, so this is for adenine, can be eliminated in one of three forms: ammonia, urea, and uric acid. Fish eliminate nitrogenous wastes through the gill filaments in the form of ammonia. And fish can do this because they are aquatic, and ammonia moves out of the gills. So say this is the gill capillaries. So ammonia diffuses out down its concentration gradient into the surrounding water. As soon as it ends up in the surrounding water, it couples with protons in the water to form ammonium. This coupling with protons to form ammonia generates a motive force that allows ammonia to continuously diffuse out down its concentration gradient. And ammonia is not permeable to the gill filaments and therefore cannot cross back into the gill capillaries. Speed is essential for the elimination of ammonia because ammonia, as we said, is the most toxic of the nitrogenous wastes. If it can't be eliminated quickly, ammonia is converted to its less toxic derivative, which is urea. Each molecule of urea requires 2 molecules of ATP to synthesize. Being still moderately toxic, urea requires a significant amount of water to dilute it before excreting it. And so if flying is your thing, urea is not an option because too much water in the body would make getting off the ground very, very difficult. So uric acid is the least toxic but most expensive form of nitrogenous waste to remove. It is so its toxicity is so low that it can be concentrated and eliminated in the form of paste. So these white droppings here, these bird droppings, is uric acid, and the black bits is feces because birds eliminate both feces and urine from the same hole. And so the white paste is the uric acid that can be concentrated and then eliminated from the body. Insects, land snails, and other many reptiles also excrete nitrogenous waste in the form of uric acid, presumably because they don't have access to much water in order to first dilute the urea. Mammals, as we will see later, including humans, of course, eliminate nitrogenous waste in the form of urea. Uric acid has a really interesting history, one that few people appreciate because we get so lost in the science of things and forget that science comes with a story that is sometimes even stranger than fiction. On many islands in the equatorial regions of South America, there are large populations of these nesting birds. And over the course of thousands of years, there has been a buildup of uric acid that is several meters thick. These deposits, called guano deposits, were once upon a time considered agricultural gold because it was a rich source of nitrogen for agricultural crops. Recall that nitrogen is part of amino acids, and amino acids make up proteins, which are the building blocks of all organisms. And so agricultural crops also need a source of nitrogen in the soil. And after just a few crop rotations, all of the nitrogen in these agricultural soils becomes depleted. So to protect yields and to ensure the security of a country, one has to continuously add nitrogen back into the soil. And so this singular source of nitrogen before the 1900s was exclusively these guano deposits. And the amount of food that a country could produce was capped by their access to fixed nitrogen. And I say fixed form of nitrogen because the atmosphere contains 78% nitrogen gas. But this nitrogen gas, these nitrogen atoms, are locked in a mighty triple bond, rendering this nitrogen unreactive. And so this guano gold was so important to the prosperity of countries that in 1856, the United States enacted the Guano Islands Act. And the act states that any citizen of the United States is able to take possession in the name of the United States any unclaimed island of guano deposits. And islands can be located anywhere so long as they are not occupied and not within the jurisdiction of another government. It also empowers the President of the United States to use military protection to protect such interests and establish criminal jurisdiction in these territories. So these armed guards shown here are essentially protecting bird droppings. But at the turn of the 1900s, two very famous men discovered how to convert the unreactive atmospheric nitrogen into our reactive form, which is ammonia. This process, Haber-Bosch process, and it was named after the two men who discovered it. And so this process changed the fate of human history. We went from 1 billion people at the start of the 1900s to 7.5 billion people roughly 100 years later. And so the guano deposits shown here went from agricultural gold to just simply bird poop. This book here, The Wizard and the Prophet, written by Charles Mann, includes one, maybe even two excellent chapters that describes the history of these guano islands. Now that we've fleshed out the importance of osmoregulation and excretion of nitrogenous wastes, we can look and compare at the diversity of excretory systems in the animal kingdom. The most basic excretory system is the protonephridia of flatworms. The protonephridia are a collection of these dead-end tubules that lead to openings on the body wall. So far, when we've looked at our little flatworms, we just thought of it as a paper flat structure with a gastrovascular cavity, some muscle tissue, and neurons. But flatworms also have these protonephridia that isn't captured in this image here. And so these blunt ends of the protonephridia are capped with what we call the flame bulb cells. And on this edge here, we have an enlarged nucleus, which is why we have this bulge towards the end of this cell. So this is the cap cell and the tubule cell, collectively called the flame bulb. On the inside of the cap cell, we have this ciliated interior that no doubt establishes a pressure gradient in order to drive interstitial fluid through bulk flow into these tubules, and the fluid then is excreted to the outside environment. Protonephridia typically just work for osmoregulation, so maintaining the osmolarity of fluids, because the nitrogenous wastes can easily be excreted easily through the skin of the animal to the surrounding water. So they just simply diffuse out into the surrounding water. The next excretory system are the metanephridia of earthworms that we find in the annelids phylum. Again, here we see a convoluted tubule, a tubular theme. And so our tubule opens up in the anterior segment, and from the anterior segment draws coelomic fluid. So this is fluid is drawn into this open-ended tubule, and as the filtrate travels throughout the tubule, nitrogenous wastes can diffuse out from the blood capillaries into the filtrate. Also, we can have an exchange of electrolytes either into the filtrate or from the filtrate into the blood capillaries in order to osmoregulate. Eventually, we get nitrogenous wastes that are eliminated here and excess solutes. And so the metanephridia of earthworms function both in excretion and osmoregulation. Next, we have the Malpighian tubules in the arthropods phylum. And the Malpighian tubules are essentially these little tentacles that extend out from the midgut of the digestive organ system and are covered with transport epithelial cells, like the ones we saw previously already, that pick up solutes and nitrogenous waste from the hemolymph into the lumen that empties into the digestive tract and eliminated through the anus along with feces. So this reminds us why an open circulatory system is so important. And so if you recall, the open circulatory system that we find in insects continuously moves the hemolymph around. And this continuous movement of the hemolymph is important in order to bring wastes within diffusion distance of the Malpighian tubules. Without this constant circulation, wastes that build up in this end of the animal will never be eliminated from the body and eventually lead to toxic levels and cause the proteins to denature and neurons to cease any signaling. Just like the metanephridia, Malpighian tubules function both in excretion and in osmoregulation in the nitrogenous wastes that are excreted in the form of uric acid. They're also excreted in the form of uric acid in earthworms. Our final excretory system are the kidneys, the mammalian kidney. The kidneys function both in excretion and osmoregulation. The kidneys receive about 21% of our cardiac output, and so each minute our kidneys receive nearly 5 cups of blood and produce 1 ml of urine per minute, which explains their very high metabolic rates. Blood is conveyed to the kidneys through the renal artery, and from the renal artery it'll split to roughly 1.2 million arterioles, and each arteriole is going to lead to a single nephron. So there are 1.2 arterioles that lead to 1.2 nephrons. There are 2 kinds of nephrons. We have the cortical nephron and the juxtamedullary nephron. The difference between these 2 nephrons lies in the positioning within the kidney. The cortical nephron, the majority of the nephron lies within the renal cortex, which is the upper portion of the kidney. So this region here is our renal cortex, and this is where we have the majority of our cortical nephrons. Our juxtamedullary nephrons extend into the renal medulla, this section here, and the renal medulla is further subdivided in the outer renal medulla and the inner renal medulla. So the juxtamedullary nephron extends deep into the renal medulla. The nephrons, as we will see, empty into the renal pelvis, and so this is the renal pelvis that is shown here. This is our renal pelvis, and this is where all of the urine is secreted, and it moves from the renal pelvis into the ureter, from the ureter into the bladder, and from the bladder out from the urethra. So we have our kidneys that empty, that process the blood, eliminate the wastes in the form of urine. The urine accumulates in the renal pelvis, flows from the renal pelvis down into the ureter, to the bladder, and out from the urethra. Looking at the mammalian nephron in much greater detail than we did with the previous excretory systems, we begin with first our afferent arteriole. Remember, I said that there are roughly 1.2 arterioles. So the renal artery splits into several million arterioles, and each arteriole leads to a network of blood capillaries called the glomerulus. So the glomerulus is a fistful of blood capillaries, exceptionally leaky blood capillaries. As blood is conveyed down the arteriole and into these the glomerulus, we have blood pressure that forces up against the walls of these capillaries, and we have a significant amounts of fluid that end up in the Bowman's capsule. So within the Bowman's capsule is we have a significant accumulation of what we call the filtrate, what eventually becomes the urine. We find the glomerulus again in this image here. So here is our glomerulus. This is our afferent arteriole. And this is our efferent arteriole. So we have the arteriole that conveys blood to these blood capillaries, and then the blood leaves these blood capillaries from the efferent arteriole. Typically, the afferent arteriole has a larger diameter than does the efferent arteriole. And so this sudden constriction here, and you see it more clearly from this image, the diameter of the efferent arteriole is much smaller than the diameter of the afferent arteriole. This sudden restriction helps to increase the pressure that's exerted on the walls of these capillaries in order to force even more fluid out into the Bowman's capsule to increase the amount of filtrate produced. If the blood pressure in here was very low, very little filtrate would be produced. So by having a much narrower efferent arteriole leading out of the glomerulus, this helps to build up, to increase the amount of pressure here. Recall that we said typically blood pressure in arterioles is about 20 millimeters of mercury. In the glomerulus, the blood pressure within these blood capillaries is about 55 millimeters of mercury. So we have significantly higher pressure, almost double, more than double the pressure than we typically find in other body capillaries because of the importance of producing more, forcing more fluid out into the surrounding. Another feature indicated on this image here is this nephron here is what we call the juxtamedullary nephron because it reaches further into the renal medulla versus a cortical nephron that sits mostly within the cortical region of the kidney. Both the Bowman's capsule on both these nephrons leads next to the proximal convoluted tubule. So it's a tubule that's highly convoluted, highly folded in order to increase the surface area. And the proximal convoluted tubule eventually dips down into the loop of Henle. So this is the nephron loop or the loop of Henle, as shown in your book. We have the descending loop of Henle, and then we have the ascending loop of Henle. And the ascending loop of Henle leads to the distal convoluted tubule, a second tubule that's highly convoluted, highly infolded, and the distal convoluted tubule empties into what we call the collecting duct. And so typically we have 6 nephrons that plug into a single collecting duct. So here we have 1, 2, 3, 4, 5, 6. So the image isn't showing all of the others because it would then not be as visible. But here we have this nephron plugging into this collecting duct, and another juxtamedullary nephron plugging into a collecting duct. And so the filtrate then flows into these collecting ducts, and from these collecting ducts into the renal pelvis in order to eliminate the urine. The same image is what we see here, except now the whole structure has been pulled apart in order to see it better. So here we have our afferent arteriole and our efferent arteriole. And our afferent arteriole leads to our glomerulus, where fluid is pushed out into the Bowman's capsule. And the Bowman's capsule leads to the proximal convoluted tubule. And from the proximal convoluted tubule, we go down the descending loop of Henle. and then the ascending loop of Henle, and from the ascending loop of Henle into the distal tubule, and then the distal tubule empties into the collecting duct. And once again, we see that a collecting duct can service 1, 2, 3, 4, 5, and perhaps 6 different nephrons. In the next few minutes, what we'll look at is the reabsorption and secretion that occurs in each one of these segments of the nephron. Within the proximal convoluted tubule, it is important to recapture, to reabsorb the nutrients that are within the filtrate. So all glucose, amino acids must be recaptured, because this filtrate here within the Bowman's capsule includes water, it includes salts, bicarbonate ions, protons, urea, glucose, amino acids, because they are smaller than proteins, so anything below protein size is able to be forced out into the glomerulus. Proteins stay within the blood capillaries, as do red blood cells. And of course, some drugs are also found in the filtrate. Up to 100% of the solutes in the filtrate can be reabsorbed back into the surrounding blood capillaries that are wrapped around the proximal convoluted tubule. So here we see everything from the bicarbonate ions, sodium chloride, potassium, nutrients, of course, this includes the glucose and the amino acids. And from the blood, so from the blood capillaries, we have protons that are moved through active transport into the filtrate, and then small amounts of ammonia that do circulate in our circulatory system are eliminated through diffusion into the filtrate, and this couples with the protons to form ammonium, and so ammonium can't then diffuse back out. So we have some elimination of secretion, I should say, of ammonia from the blood capillaries into the proximal convoluted tubule. Everything else from the filtrate is generally picked back up and returned back into the circulatory system. In the descending loop of Henle, the only movement that we have is the movement of water into the surrounding outer medulla and inner medulla. And the movement of water through osmosis is possible because the outer and inner medulla has a very high solute concentration. And so the osmolarity here, the osmolarity of the interstitial fluid here is very, very high. And because it is very, very high, this helps to draw water out of the filtrate and into the surrounding renal medulla. And we see this movement possible if we contrast it with this image here. The osmolarity of the filtrate in the proximal convoluted tubule is 300 milliosmoles per liter. 300 milliosmoles per liter. This is the same osmolarity as our blood. The osmolarity of our blood, as shown here in the glomerulus, is 300 milliosmoles per liter, and the osmolarity of the filtrate in the proximal convoluted tubule is also 300 milliosmoles per liter. As the filtrate leaves the proximal convoluted tubule and moves down the descending loop of Henle, and water moves through osmosis into the surrounding high osmolarity environment. And so if we look here, the osmolarity here gets increasingly high from 400 to 1,200. So we have the net movement of water into the surrounding interstitial fluid. As the filtrate continues down the descending loop, the osmolarity of the filtrate increases to a staggering 1,200 milliosmoles per liter. As the filtrate moves back up, we have now a biological membrane, epithelial cells that are impervious to water. Water cannot cross through the epithelial cells lining the ascending loop of Henle. This is why here we have absolutely no water moving out from the ascending loop of Henle into the surrounding inner and outer medulla. Instead, we have sodium chloride that moves out, diffusing down its concentration gradient, because the concentration gradient of sodium chloride here is not as high as it is inside this tubule. So initially we have diffusion through passive transport into the surrounding inner medulla. As the filtrate moves up, we have active transport pumps that are actively moving, so in using ATP to move sodium into the outer medulla. This process is crucial in order to continuously ensure a high osmolarity environment here, in order to ensure water is recaptured and returned back into the circulatory system. As the filtrate moves into the distal convoluted, the fine-tuning of the composition of urine is achieved by the distal convoluted tubule. This is where the body can osmoregulate and reabsorb sodium chloride or eliminate potassium. Also, this is where calcium can be recaptured from the urine if blood calcium levels drop. And so we will see this next class when we talk about the endocrine system. And so when blood calcium levels drop, because calcium is a crucial electrolyte that enables neurons to generate action potentials, the distal convoluted tubule can control the amount of calcium that is eliminated in the urine versus how much is recaptured, which means reabsorbed out of the filtrate and returned back to the circulatory system. Remember that both the distal convoluted tubule and the proximal convoluted tubule are both in intimate association with blood capillaries. Once the filtrate enters into the collecting duct, water there's another opportunity to recapture water, except this time this movement of water is under hormonal control, and it's based on the osmolarity of your blood. If the osmolarity of your blood becomes really high because you just ate a bag of very salty peanuts, your body will allow more water to be recaptured from the collecting duct and returned into the circulatory system to very quickly lower the osmolarity of your blood. From the collecting duct, we also have sodium chloride that is moved through active transport into the outer medulla, again with the intention of generating a high osmolarity environment. And also we have urea, which weirdly also moves out through passive transport, so it diffuses out of the collecting duct into the inner medulla, again to generate a high osmolarity environment. This is why we call it a two-solute model. Two solutes, sodium chloride and urea. Both these solutes create a hyperosmotic environment that continuously ensures that in the descending loop of Henle, we always have water that moves out. Otherwise, we would lose too much water through the excretory system. Now, with our blood vessels in intimate association with the proximal convoluted tubule and the distal convoluted tubule, and all of this water exiting from the descending loop of Henle, much of it is recaptured from another set of blood capillaries that are in intimate association with the loop of Henle. And so these blood vessels here, shown here, are the vasa recta. So the vasa recta are important for recapturing all of this water. So much of this water is returned. And the flow of water here is important to note. The flow of blood, sorry. So the blood flows down this part of the vasa recta, and as blood flows down this side, this way of the blood capillaries, the osmolarity of the blood here goes from roughly 300 milliosmoles per liter to down here where the osmolarity of blood is roughly 1,200 milliosmoles per liter, which is the same osmolarity as the surrounding interstitial fluid. But luckily then blood turns back up this way, moving down the concentration gradient of the renal medulla, And so as the blood moves back this way, it returns back to 300 milliosmoles per liter. Eventually, this blood capillary leads to the renal vein, and from the renal vein it exits the kidneys with osmolarity reestablished of 300 milliosmoles per liter, and all of the urea nitrogenous wastes are now eliminated, and electrolyte homeostasis is achieved. In this final slide, we have another really beautiful image generated by a very talented medical illustrator. So this is, if you notice, the first image that I showed that was drawn by Yuxian Ren. She's a former student that had taken this course many years ago, and now she has become a very talented medical illustrator. So those are her images, and I've used a couple already in this course. This is another one of those really beautiful and sort of artistic rendering of biological processes. This is what she calls here a nephron forest. So in this nephron forest, we have our convoluted tubules, our distal and proximal convoluted tubules that plug into, so the distal convoluted tubules plug into these collecting ducts. So here we have our collecting duct. We also have the loop of Henle shown here with the vasa recta in intimate association with this loop of Henle. So a very beautiful illustration of the mammalian nephron. Also indicated here is the different types of epithelial cells that we find throughout the nephron. So the proximal tubule typically has simple cuboidal epithelial cells because these cells need a lot of mitochondria to produce much of the ATP necessary for the energy-intensive reabsorption process. So the reabsorption of the nutrients and the reabsorption of other substances from the filtrate. Both the descending and ascending arms of the thin loops of Henle. So notice that it goes from a thin segment to a thick segment. So we have a thin segment here and a thicker segment there. So the thin segments are composed of simple squamous epithelial cells, and they're not metabolically very active, and they don't participate in active transport. But the thick segment of the ascending loop of Henle, recall, is moving sodium chloride actively, and so the epithelial cells need to be studied with pumps in order to move the solutes against their concentration gradients. So the thick ascending loop of Henle is linked with thicker simple cuboidal epithelial cells because it's involved in active transport of sodium chloride and even potassium. Now we have the distal convoluted tubule that again has simple cuboidal epithelial cells that is similar to the proximal convoluted tubule, except the proximal convoluted tubule also includes a brush border, as we saw in the transport epithelial cells of the salt glands of the marine birds. The simple cuboidal cells of the distal convoluted tubule don't have this brush. Finally, as we move from the distal tubule into the collecting duct, it seems that the epithelial cells move from simple cuboidal cells to simple columnar cells. You're not responsible for this slide in particular, where I discuss the different types of epithelial cells that we find in the different regions of the nephron. I've included it because it helps to illustrate the relationship between form and function and the importance of having the right cell shape in order to achieve the right function. And because each region of the nephron has vastly different functions, it's a nice way to signal that nice relationship of form and function, but you are not responsible for this slide. So next lesson on the endocrine system, we will also cover the hormonal controls of the excretory system. And so I'm moving that section into next class.